It's my pleasure to introduce Dr. Purevjav for today's seminar. Dr. Purevjav is Associate Professor of Pediatric Department in our campus. She got her MD degree from Leningrad State Medical Pediatric Institute, Russia in 1989. She did her internship and residence in National Medical University of Mongolia from 1989 to 1992. She did her fellowship and pediatric cardiology in St. Petersburg State Pediatric Medical University, Russia from 1992 to 1994. Then she got her PhD degree from the Shimane Medical University, Japan in 2003. She did her postdoc training in Baylor College of Medicine, Houston from 2003 to 2007. She became the instructor in Baylor College of Medicine, Department of Pediatrics, Houston in 2007. She got her assistant professor position in Cincinnati Children's Hospital Medical Center, Department of Pediatrics in 2009. She became associate professor in Pediatric Department of UTHSC since 2015. She has published more than 30 science papers for the last 20 years, and she got her own grant funding last year. The title of her talk today is Utilization of BXD strain resource for genetics research of human cardiovascular disease. Now let's welcome to the Dr. Purevjav.
Okay, thank you very much for the introduction. So let me get to the, where is the Zoom? Okay, let me get my, I need to share my screen, yes?
Yes.
So to the, okay, here we go. So what is this? Interesting. When I get the desktop, there are so many desktops. Here we go.
Yes, yeah.
So do you see?
Yes, you can see.
Share screen. Okay, so,
yes.
Okay. Thank you very much for the introduction, and thank you very much for the Department of GGI for the invitation to present some work that we did with collaboration with Dr. Lulu and Rob's group. So, and today I would like to present some of the data, how we utilize BXD strains resources for genetic studies of human cardiovascular diseases. So, and first, I want to just say a little bit about our research lab, although we are on the same floor, and we didn't see for for more than a year. So I'm just want to remind that we are mostly working on the cardiovascular diseases, including cardiomyopathies, arrhythmias, and heart failure in humans, and exploring genetic basis of that diseases. And also we're exploring pathogenesis, molecular and pathophysiological mechanisms, how these disorders develop, and we want to find etiology or genetics or mechanism-oriented therapies for these disorders. So the research lab maintains 3 repositories, and there are human repositories. The very first repository is a pediatric cardiomyopathy repository, which was established in 1980s by Dr. Tobin, who is my long-standing supervisor and the mentor. And we do have more than 8,000 human samples from blood, DNA, as well as the cell lines, including iPS cells, and from more than 3,000 patients with cardiomyopathies. And this will also have samples from their relatives and the control people. So, and this is one of the biggest, pediatric cardiomyopathy repositories in US or in worldwide. So, and we also have a report by banking of the North American arrhythmogenic cardiomyopathy. This is NIH funded repository. We have samples from more than 600 patients from for now. And we have very, like young, let's say, Labona cardiovascular specimen repository, which we run last 4 years. And we're collecting blood samples. We're collecting the human heart tissues, explanted heart tissues, and we're also, creating cell lines for those, patients. So, and most of these repositories are multi-center studies, and most of them NIH funded. So, and besides that, we do the cellular and mouse models, and as well as we're doing the recently cardiac phenotyping of the murine genetic reference population of BXD mice that Rob and Lou have in their lab. So, and because we are working on a cardiomyopathy, I would like to give a little brief characterization of what the cardiomyopathies are. So cardiomyopathies are the cardiac diseases and diseases of the heart muscle, and it's a devastating disease, and it's one of the leading causes of cardiac related morbidity and mortality in a pediatric and adult population. There are no effective treatment is established so far, and nearly 40% of children with symptomatic cardiomyopathy receive heart transplant or die, unfortunately, within 5 years of diagnosis. So there are 5 cardiomyopathy types are distinguished by American Heart Association recently. So it's from 2006. There is hypertrophic, dilated, restrictive, non-compaction cardiomyopathy, and arrhythmogenic cardiomyopathies. Why I'm expressing on these types of the cardiomyopathies, because the BXD strains display almost all of these types of the cardiomyopathies as a phenotypic traits. So hypertrophic cardiomyopathy is a cardiomyopathy when the myocardial walls are thick and the volume is small, and because the volume of the chambers are small, they express diastolic dysfunction. In contrast, dilated cardiomyopathy have a very thin ventricular walls, but the chamber is big or dilated. That's why the name come as a dilated cardiomyopathy. Restrictive cardiomyopathy has a restriction of the muscles. It cannot extend. And that's why, and it's kind of like, trapped within the pericardium. It cannot, relax sufficiently to pump the blood. So, and the non-compaction cardiomyopathy is characterized by this non-compacted spongy type of the myocardium, and usually they do have recesses and trabeculations and causes the failure of the heart. And the last type of the cardiomyopathy is arrhythmogenic. Name says everything. There are rhythm disturbances from supraventricular or ventricular tachycardias, and that causes cardiac remodeling and heart failure. So I just put this, the cartoon just because, just remind that the muscle cells are the mononucleated. It could be bifurcated, and it is striated because there are some there are Z discs from one membrane to another Z disc, making the the name of the striated muscles. And it's between the cells, we have intercalated discs that connects one cell to another one, and and we do have a lot of, we will talk why I'm talking about the intercalated discs, Z disc and membrane of this, because, genetics comes from location. So next, so going to the genetic basis of the cardiomyopathies, different cardiomyopathies have different genetic causes or gene mutations or genetic abnormalities in the different genes. However, same gene can cause different cardiomyopathies. Like, for example, if we look here, I'm sorry, so if we look here, the genes that I put here in a circle, the genes we are focusing in our labs. For example, CSRP3 that encodes muscle-linked protein, which is the transcription factor, is responsible for development of DCM, is responsible for RCM. So it could cause any type, these two different types of the cardiomyopathies. For example, Nibulet gene that we also focusing can cause left ventricular non-compaction on top of the DCM and HCM. So the message is cardiomyopathy has a very complex genetic basis, and it's it is very, very difficult to distinguish what's the causative genes, what's the modifier genes, because of the this complex inheritance patterns. So it means that the establishing genetic reference population for cardiomyopathy traits is very important for translational relevance and for the in animal modeling as well as in human research. So the BXD is very valuable because they are bred under controlled environment, and we can see the true genetic basis of the cardiomyopathies by studying the genetic gene expression in these traits. So and I just put some BXD family recombinant inbred strains, why it is important, what is the the situation for the, so this is the very good source for us to look at for the cardiovascular traits. So in this study that I want to present, we wanted to establish cardiovascular phenotyping parameters across at least 40 strains, but we overdo. We did 44 strains, and we analyzed more than 400 mice, male and a female, 5 per sex, and we used 5 to 6 months old in BXD mice for the experiments, such as echocardiography, electrocardiography, or ECG, blood pressure, cardiac histologies, and we use cardiac microarray analysis for the genotype, phenotype association analysis and QTL mapping. So I will cover echocardiography and electrocardiography in this presentation because we do have so much to do, and we just split it to in this for this time. So okay, so before we go to the cardiac traits, I just put very simple cartoon how we are distinguishing the hypertrophy versus dilation. So on the left top, you see the normal heart. And when we do the short axis cut through the ventricles, we can get the donut type, like the view of the left ventricle from the top of this view. So, and usually, you know, the echocardiography is done through the long axis and short axis views. I'm sorry. So, and what we're distinguishing LV volume, which is the, you know, black, white, you know, circle, and we distinguish myocardial wall, and we measure it, and we just, you know, give the, you know, what's the, you know, the values of this. In hypertrophic cardiomyopathy, what we see, because of the, you know, hypertrophy of the muscles, we do see the thickening of the walls, but we have the decrease in chamber volume, which, you know, causes diastolic dysfunction. In a dilated cardiomyopathy, in contrast, we see the thinning of the walls, but the increasing the, you know, chamber volume. That's we will, you know, look at here. So first we look at on a heart rate. So the black arrows show the two parental strains. I don't know where is the another one here. So, and the heart rate is variable among the BXDs, but we cannot distinguish, is it because of the dilation or because of the hypertrophy? Because on the both situations, we do have increase of the heart rate. So we, you know, saying that, okay, heart is enlarged. To distinguish what is the, you know, causes the heart, the weight increase, we look at on the LV volumes, and we found the differential values among these in male as well as a female mice. If you look at them here, the black arrows in the C57 has these values, but we have other, you know, values under the red arrows showing bigger volume at systole and a bigger volume at the diastole. If you look at on the left side, LV volume at D and LV volume at C, showing this C is the systole, D is a diastole. Systole is the when the heart contracts, and diastole when the heart is relaxed. So, and all the, you know, BXG strains starting from 113 has a LV dilation, which is significantly different from the C57 B6 control, which is, you know, is the sign of the dilated cardiomyopathy. So, and I also, you know, put LV restriction. What does it mean? This means that the volume during diastole, during relaxation, that the muscle is relaxed, and we should have maximum volume for preparing the pumping the blood. The heart is restricted, which means that these strains, starting from 65B to the 74, has a significant difference in the LV volume during diastole from the D2 mouse or in a BXD6, which is the control, saying that this strains could have left ventricular restrictive cardiomyopathies, which we are, you know, we are going to, you know, which we will, you know, look at on a different, you know, different parameters to see if it's, you know, fitting to the restriction. But this is the very, you know, first sign of the, you know, restrictive cardiomyopathy in this. So same thing in the female mice, female BXDs, we also have the variable volumes during systole and during diastole, and we see the, you know, we can distinguish which strains have a LV dilation and which strains have LV restriction during diastole. So, and then we performed QTL mapping for echo traits, which is in the case is the LV volumes, and we found the significant, you know, the on the QTL in a chromosome 8. And this, chromosome 8 had another, had the positive correlation with the left ventricle internal dimension. It's a diameter of the left ventricle, and this is the volume of the ventricle during diastole and during systole, and every single the trait that shows dilation of the left ventricle is correlated with the gene, which is in NDRG4, and we found that this is the candidate genes for the dilation of the left ventricle through the BXD strains. So when we, next we looked on the left ventricular posterior wall thickness, as I showed that this, the wall thickness will give you the signs of the hypertrophy, true hypertrophy. So although the heart rate can be increased, but it could be increased because of the dilation. That's why we are distinguishing. Is it because of the dilation or the hypertrophy? So left ventricular posterior wall thickness was also very variable, highly variable among the BXDs. So, and I highlighted here in red the LV hypertrophy in these strains starting from 44 going through this, and these are the strains that had significant difference compared to the other two controls in the posterior wall thickness, in a diameter, in diastole, as well as in males and the females. So these are the the strains that could have left ventricular hypertrophy among the BXDs. So, and we did the QTL mapping for the LV posterior wall thickness, and we found chromosome 7, which is in the locus on the chromosome 7, which was significantly correlated with the LV posterior wall thickness. So, and when we looked on the candidate genes on this chromosome 7, we found several genes that are, having non-synonymous SNPs among the BXDs. Most of them have, and one of the genes, just D2, has splice site mutation, and it was regulated, found to be cis-regulated. So this is, we're thinking that's the candidate genes for the cardiac hypertrophy based on the association of the LV posterior wall thickness to the gene expression, cardiac gene expression. So next, heart function, very important, and we looked on the heart function, and we looked and we did the QTL mapping among the BXDs. So before that, I'm just wanted to make sure that we are understanding that everybody could understand simply what is we are looking for in terms of assessing the cardiac function. So we are looking on a diastolic relaxation, and we're looking on a systolic contraction, and the variance between these two will give you the true function. So, and we look on cardiac function through the two variants, the variables. It's first is the ejection fraction. Ejection fraction gives you what is the percentage of the blood is pumped out of the heart, and you can calculate it by differences of the stroke volume, SV. It's called stroke volume. It's a difference between the volume of the left ventricle at systole and diastole. and you do the, the differences into the end diastolic volume, and you multiply 100 times, and you get, percentage in the ejection fraction. Fraction shortening is it's a similar thing, but it is, calculating the differences or, percentage of the, pumped, or, pumped blood through the diameter of the ventricles. And for example, if you look at on the EDD, it calls end diastolic diameter of the, of the chambers. And similar, the design, and it both shows you the, the how much blood is pumped from the heart during the, contraction. So if you look at on the, EF in NFS in the male BXDs, we also found almost twofold variance among the BXDs. So if we look at on the black arrows, that shows two controls. One is the control in DBA D2 mouse. D2 mouse has a high EF. D2 mouse doesn't have cardiac failure or, the impaired cardiac function. It is hypercontractile, means the function is very good because it has the hypertrophy of the muscles, and hypertrophic muscle the pumps blood very well. So, but in some of the strains that comes from the crosses of the D2 and B6, we do have the strains that have significantly impaired systolic dysfunction, and it's number 34, 9, and 52. And they do have significantly reduced ejection fraction, that's showing that this, strains have a cardiac function. So in the females, we have more strains that have systolic dysfunction, but we have also the strains that also gives you the hypercontractile function, like 49, 72, which have the hypertrophic phenotypes on based on posterior wall thickness. And starting from 61 in the females, we have significantly reduced ejection fraction, which shows the dysfunction, systolic dysfunction in these strains. So again, we did the QTL mapping for echo traits, and in this case for the fraction shortening in ejection fraction that shows the function of the muscle, and we found the significant QTL on the chromosome 3. In chromosome 3, we looked on the further, and we found many genes that are, located on the chromosome 3 and have, for example, top 4, 5 has a SNPs, non-synonymous SNPs among BXDs, and we have the cis-regulated genes that, among all these. And importantly, some of the genes like troponin 3, VARS2, and I think I don't see that here, but has the cardiomyopathy associated cardiomyopathy associations previously reported by others. So which means these are genes could be candidate genes for the function, LV function and systolic dysfunction. So next, if you have any question, you can interrupt me and ask on the echocardiography things. And then we did echocardiography tracing in the BXD mice, and we look at if there is some disturbances of the cardiac rhythm there. So because there are many students and and most of the, the participants are not, cardiac people. I just wanted to make sure that, we show how we distinguish rhythm disturbances in these mice. For example, on the top, on the BXD55, you see the three, ECG signals. So when the heart contracts, it gives the big, the spike of the, the electrical signal that you can see the spike going up. But when you do the 1, 2, 3 spikes, which is we, count as a normal rhythm, you are going to have another, rhythm that is totally different. It has a totally different, appearance. So this is, we call it the ventricular contraction, and it's the premature. Premature means it should not come here. So, and usually, this comes from the atrial contraction, and then ventricles contract, but this is, contracting in a different way, and which means this is not coming from normal way that we are expecting, and we call it PVC, okay? So it calls premature ventricular contraction. The same pattern you see in a BXD68, same pattern you see in BXD18.3, and you see also 78, and we also have some like atrioventricular blocks. What does it mean? When you have 1, 2, 3, 4 normal in a BXD48, and then suddenly you don't have this ventricular normal signal, and it's a dropping, and we call it atrioventricular block, okay? So, and there's, PACs that comes from atrium. Premature atrial contraction will give you like, in 169. This is just examples how we, distinguish all the, PVCs and PACs. So it's a very important that, we are knowing what on them, what we are looking for. So if you have this, different, the different appearance of the ventricular contraction, and it's more than 3, in a cardiology, it is considered as a tachycardia, and it is considered as a ventricular tachycardia that is, abbreviated here as a VT. And it's a serious thing because VT is one of the major causes for sudden cardiac death. And the B65B and 65F both has ventricular tachycardia, which is, I think it's a very important, sign that we can pursue the studies of the BXD mice for the arrhythmogenic ventricular cardiomyopathy genetic studies. And there is, a lot of, the PVCs again. And if you look down, BXD48F has the I'm sorry, the 66F has a 6 sinus syndrome signs. So which means the sinus syndrome doesn't work. And you have the block, very long block, which blocks at least like 1, 2, 3, cardiac rhythms in that. And usually, it is one of the reason of the syncope or the sudden cardiac death as well. So we didn't do much, yet, the genetic studies or genetic association of this yet, because the number of that we looking in this is not, let's say, enough or sufficient to do the, QTL mapping. Probably we'll do the more, the ECGs in the BXDs to have a statistical significance. However, we have different, the different cardiac disturbances, rhythm disturbances in these mice. For example, VT, SSA, or atrioventricular blocks in this, which is, I think it's very important. And also, we are doing measurements of all cardiac signals, Ecg signals in these mice. And these studies are underway. We have it is a lot of the analysis, and we are in a progress doing the association studies using the ECG, other parameters of the ECGs. So to use or utilize all the BXD sources in studying human cardiomyopathies, we are very eager to do it because the genetic basis in human cardiomyopathy remains unclear or imprecise in most of the cases. For example, 60% of the human cardiomyopathy cases does not have any genetic explanation, despite there are so many, so many groups, so many, labs doing the whole exomes, whole genomes, and there are so many, efforts finding the genetic basis, and we still are struggling. So, and it is because of the complex traits, and because of polygenic nature, and because of the various environmental factors. So, and I think the advantages of the BXD sources for human cardiomyopathies is very important, and it's very significant. And in addition, the D2 mouses, which is the parental strain, is determined as a natural model of the cardiac hypertrophy. And there are more than 6 million SNPs, insertion deletions, and thousands copy number variations segregate among the BXDs, which is similar to human population. So similar to the BXD repositories, we do have human repositories. We work with many clinical sites. It's almost like more than 20 clinical sites across the United States and Europe. They're sending us the DNA or blood for the collection of the genetic material for genetic studies. So there is clinical sites doing clinical evaluation, doing the ECG echo like we are doing in the BXDs, and they do cardiac catheterization treatment, the cardiac transplant, including. So, and they all send the clinical data to the New England Research Institute or University of Rochester to deposit the clinical data. And we work with this clinical site as a genetic core labs, one of the labs, and we're doing biobanking, next generation sequencing, next generation whole exome sequencings. And we're also, doing the biomarker studies, including the plasma biomarkers for the cardiac diseases. So this is, would be very, very valuable for this, human studies if we can employ the BXD resources for understanding human diseases. So I would like to present one of our families that we've been studying since last century. So it's a big, family, came from Europe, seems. So, and because this family has divided into even 3 continents, like America, Europe, and Australia. And so many studies were done in 2000, and they did, the linkage analysis, and they linked the genetic basis or causes of this with the chromosome 10, Q21, 23. There's many genes, and In that time, they did direct sequencing of the, I think, more than 20 genes in these regions, and they couldn't find any of the, mutations or variants that could be causative for this. When the whole exome sequencing became available, we did whole exome sequencing in in 10 family members, and we found 3 candidate variants that could be a cause. So we found KCNM1 variant, we found XO11, Axilin 11, and DYDC2 variants that are all located on 10q2123. So this is the pedigree of these big families. And when I put all these pedigrees, and I put all this, the plus minuses with, with 3 genes, I could partially associate clinical signs with the clinical severity and with the clinical, the onset of the disease. And we found that, okay, this is not all doing because of these 3 genes. For example, if you look at on this guy, the 312, 3rd generation patient number 12. This index patient has all 3 variants. However, it is not same as her siblings, like number 16, number 17, which is which is a carrier of all 3 variants. And then when we go to the proband, which is indicated by the arrows on the top left, generation 4, patient number 13. This patient is a carrier only of 2 genes, but it shows whole spectrum of the the phenotype, which doesn't correlate with. It is not the mutation burden. It is something different that is not only on the 10q21. In that case, the BXD resources helped a lot. What we did, we ran all the variants that we found in this family through the BXD cardiac gene expression, and we looked on the association of these 3 genes. For example, between, if all 3 genes have an association between of all this. And we found that we ran association between all these 3 genes, and we ran the associated genes outside of the 10q21. So, and we found that the variants in the titin, variants in the chorin or TRPM in generation 3, 12 affected patient has the associations with a KCMN1 or AXA1. So, and which means there are the genes that are associated with these 3 genes that are located on chromosome 10q21 and maybe affecting on the severity of the cardiomyopathy phenotypes or the onset of the cardiomyopathy in these pedigrees. For example, proband, who is the first index patient, comes to the clinic. She carries only 2 variants, but she carries chorin variant that is associated with the KCMN1 and AXA1, and it could be the reason that we are dealing with the different phenotypic expression among the family members that carries different causative, let's say, variant burden. Now, in the genetics, the field, we're not telling mutation, we're telling mutation. We're not telling mutation, we're telling variant, because the variant expressivity, variant the severity can be affected by many complex, the genetic and non-genetic factors. That's why, all the mutations are called variant. It could be pathogenic, it could be non-pathogenic, it could be benign, or this is a variant. So let's go. Okay, so if we look at on the on the chromosomes in these families. So this is the chromosome 10 that carries 3 different variants on the locus that was identified by linkage analysis, which is associated with this DCM in this family. However, our genetic association studies through the BXDs found different, or found many variants that could be important that influences the cardiomyopathy phenotype in this family. For example, all the variants identified by Asterix are novel. For example, chorin, titin, and the TMRPMs seems like they are novel variant, but we found it through the BXD association studies in these families. So and that's you. Okay, let me get the, okay. I don't know why it is not working on my side, but I cannot, get the, what's going on here? So let me my computer is, do you hear me? Hello?
Yeah, we can hear you just fine.
Okay, okay, I'm sorry about that, but my computer is let's say, the
so
frozen. I'm sorry.
Your mouse works actually. We can see the mouse.
Enkhe, we can see your computer. Take your PowerPoint slide and drag it into the chat box, and then I can run it for you. So take your PowerPoint slide deck.
Oh, yes. Oh, okay. It's run. Okay,
There,
now
there
it's good. Okay. So here's the conclusions of my talk. So I wanted, I didn't go through all this, very let's say complicated echocardiography, ECG things, just not to confuse students and confuse non-cardiac people, non-cardiac collaborators. So, and from our studies that we've done in the BXDs, we can say that echocardiography traits associated with cardiomyopathy are inherent from D2 mouse, and they are segregated among BXD families. And these echocardiography traits are very among BXD lines, and we can distinguish multiple associated QTLs that demonstrating that BXD family can be used for genetic or systems genetic analysis in the cardiovascular systems, the analysis. So, and the BXDs are suitable as a GRP to identify genetic factors and modifiers that influence cardiomyopathy phenotypes. And we also have examples how we successfully use the BXD resources to discover novel genes associated with cardiac diseases in humans. So, and I would like to acknowledge my collaborators and my, the lab members. So from Heart Institute. So for especially Jeff Tobin, who is providing long-standing support for these studies, and other members, previous and current members of the labs. And particularly, I would like to express my the thanks to Rob, Lou, and Fui for the for the opportunities to do this job. And also, that I would like acknowledge the Cincinnati Children's Hospital, especially Dr. Zhang, who did the whole exome sequencing in our human samples. And I put a lot of our R01 grants, and this is the grants that supported for establishing and maintaining the human repositories as well as mouse repositories of the BXDs. So if you have any questions, please, I'm open for for all.
Thank you very much for your great talk, the Enkhe. Yeah, you connect the mouse and the human cardiovascular disease. That's great example to use BXD mice resource. Yes, yeah. Thank you.
Thank you. Any questions? I'm happy to answer.
Enkhe, there are a bunch of questions in the chat that I put in there.
Oh, okay. I'm sorry that I'm very bad. Actually,
but I'm hoping some other people will ask questions too. Because I agree with Lulu. This was a really good presentation of how to essentially start from scratch using to do the alignment of human genetics on any disease and mouse genetics on any abnormality or just even normal variation.
Yeah, thank you so much.
That's been doing the same kind of work for stroke models. Wenjing's doing the same kind of analysis for regenerative medicine and blood vessels. So a lot of upside here.
Yeah, I think it was very, very effective. Let's say collaboration with a human from bringing together human and mouse, repositories, and there were a lot of the holes in our human studies, because we cannot have number 2 mouse or number 2 individual to do the significant the association studies and see the significance between the trait that you see in a human and the genetic the variants. And BXD research is very valuable.
One of the nice things about your phenotype, Enkhe, is that it's possible to get long time series data in the same way that it's possible to get long EKG time series data for humans. And Bob Davis has been doing a lot of machine learning to see to what extent he can predict diseases that you would not expect. For example, you can take the EKG in humans and you can do a pretty good job predicting Parkinson's disease in individuals who might be at risk. So it'd be fun to add the time series data. So how long are your EKG recordings for the mice?
Yeah, we do 5 minutes for each mouse. And because, we don't have Holter type, the things, maybe, we need to, purchase Holtering, because you can do, 24-hour EKG in the mouse. So, and it is doable, actually. It's a very, it would be very, very good, let's say, investment to look at on this, because only on the EKG that we are, monitoring for 5 minutes wouldn't, show you whole spectrum of the rhythm disturbances in the mouse. And what we can do, we can, do the initial 5-minute ECG, and if we find something, we can just, go and just put the mouse on a 24-hour monitoring. And it is doable. It is doable in mice, and it's very important.
How do you acclimate the mice to the EKG? Is it their first time being recorded, or do you run them a few times before you do the real recording?
Okay, so the ECG recording in mice in our lab is in anesthetized mice. So we just let them sleep first when we do, because it would, you know, interfere on the recording if it mouse is not. So there are some, you know, let's say differences between in the conscious and non-conscious muscles in terms of the ECG recording. So some people say, okay, conscious mouse will tell you, will give you more, you know, ECG, you know, like the rhythm disturbances. So and the 24-hour monitoring is the, you know, conscious mouse monitoring. It would be, you know, very interesting to see if there is, you know, significant difference between conscious and non-conscious, like, you know, mice as well.
You didn't mention mapping just plain old heart rate. Is that because you don't think heart rate is a good measure?
It is okay. It is okay to see the, you know, association between heart rate and the, but because, you know, we are doing the ECGs and echocardiography in anesthetized mouse, it would not be, let's say, real, you know, the numbers. That's why we don't do the association between heart rate and the, you know, the genetic, you know, gene expression in that case. So yes, you know, if we can get the 24-hour monitoring, Holter monitoring in the mouse, that would be, you know, very interesting to see if there is an association. It should be some, because, you know, we do see the bradycardia in this, you know, mice, some of the mice that giving you the six-signer syndrome, AV blocks. It should, you know, interfere on the, you know, heart rate in these mice. Okay, so I'm going through the, you know, the messages. What do you get when you map heart rate? Okay. Oh, I already did that. So be cool to add the aging analysis on high fat diet with C and increase the heart disease in female BXDs. Yes. So that's the, you know, the maybe our next step to introduce environmental factors like high fat diet, or maybe, you know, low fat diet, or other, you know, stressors like, you know, running, which, you know, we propose in our, you know, like grant this time. So this is, would be very interesting to see in the BXDs and to see if these, you know, environmental factors will influence the phenotype and genotype association in the mice. So real opportunity here for machine learning. Oh, we already did that, you know, so mouse cohort to human families, to mouse cohort, That is translation genetics and genomics. I think it's a two directional thing. And now they say from bench to bed, and then, you know, they say, okay, there would be bench to bed. So I think it's a two directional thing. And it's, you know, I think the BXD cohort is very valuable on that case, because the most of the genetic studies that are done on a genetically engineered mouse, it's done on a single background. And it's sometimes you see conflicting data from different labs, because different labs make their genetically engineered mouse on a different background. For example, we like BXD background because not the B6 background, because B6 gives you the more, you know, and more cardiac phenotype. And then most of the labs use the B6 background, but some labs use different, you know, background, which gives you totally different, you know, the phenotypic expression. So, and that was the one of the reasons that we were so interested in phenotyping, cardiac phenotyping of BXCs, that we will get this whole, you know, reference that all labs, or maybe numerous labs, can just reference to our, the data.
Yeah, that's the key thing. It's very important. Yeah. So you have found the abnormal symptoms from your heart functional examination. So it's measuring, for example, from the EKG, you do see the waiver is abnormal. So do you think some BXD mice can be used as the cardiomyopathy animal model?
Yes, I think so. But we need to let's say this way. So cardiomyopathy is a syndrome. Let's say it's a clinical syndrome connected with the histology, with the other the laboratory exams. So which means we have to complete whatever we started. So we did. This is the very beginning steps of the cardiac phenotyping in the BXDs. So after ECG, echocardiography, we have to do a lot of histologies showing this, phenotypic expression in the clinics. Has a lab or, molecular or cellular or, the organ level remodeling that will support to the clinical disease. So which we're doing right now. And also some, important, the signs of the cardiomyopathy is autophagy, is apoptosis, is a necrosis that showing that the muscle cells undergoing some kind of the death or disease or necrosis or damage. That's we have to do in the next steps. That is, it would be a long story, let's say. Not yet.
Yeah, I think that would be a great thing to do. And you should talk to David Ashbrook and Amandeep Bawa about mitochondrial function in heart, because heart is so energetically, well, so chock full of mitochondria, obviously. It's has by far the highest metabolic demands of any tissue in human or a mouse, probably even higher in a mouse because of the ridiculous heart rate. But you'd expect kind of an age gradient in mitochondrial function due to high ROS damage in cardiac muscle. And we have aging heart, and we could do mitochondrial assays. I think David may actually be doing that as part of his new grant application that appears to have just gotten funded. There's so many ways to take this, Enkhe.
Yes.
You phrased it in the context of cardiomyopathy, but we know that cardiovascular disease is the major killer of aging humans.
Yes.
And as you point out, of some pediatric cohorts. But it's the aging that really gets the heart. And it's the aging and the diet and the stress level. And these are all just great problems for an aging study along the lines that you've been doing. And you can't touch anything. Nothing is as good as the BXDs at this point for this kind of G by E study of just normal cardiac function with age, with diet, with stress levels, with ethanol, whatever. We have just so much data. So I really encourage you and Lou and Jeff and team to keep going. The other thing you have going for you is the deep human data, which is unrivaled. I mean, Jeff and you have been working on this for a decade or more. Probably in Jeff's case, maybe 3 decades.
Yeah, I think in the 4th decade, I guess, because he started from 80s.
It's amazing. We could be in a world-class, world leadership position for mouse human cardiovascular disease translation.
Yeah, exactly. So it is, it's a very good, let's say the, bond between human and, mouse, the repositories, and I think all the aspect that you're told, it's a very important. We have to, just, pursue all this, and I'm happy that, we did this, initial phenotyping in this mice, and it could be, this, the phenotyping can be used for comparison with aging mice, which we are, maybe, we will pursue someday, like when we get many more people doing that.
Robot
So,
have aging colony also treated by the high-fat diet. So if you have the poster, you can do the heart function measurement, just like EKG from our mouse colony.
yeah.
See
There's so
Byron,
much to do.
on the our participants, but I do see Wen Jing there, and she's got a lot of background in metal concentrations in heart and the pathology associated with heart disease and metal load. So has anybody done the metals in the aging BXD heart yet? Wen Jing, have you?
I have collected some heart tissue and not yet.
From aging mice,
Yeah. Dr. Lu gave me some heart tissue, but that just doesn't have time to measure the metal concentration yet.
Well,
but the tissues are there.
Wen Yan was on there too, so make somebody in Byron's lab do it for you.
Yeah.
Okay, Byron.
Yeah, there's so many, we can, find in aging mice. So it would be very, very important to look at on the cardiac function, as well as on the, this all, parameters by echocardiography, and connect with all the environmental factors. This would give us a lot of information. Yeah. I believe in it. So
has anybody
now it is just the first step that Robert will just establish the baseline data for the heart function for about 40 big strength. Next step, of course, we can compare with the high-fat group, aging group, and exercise group. And then a lot of granted the opportunity there, projected there.
I would encourage all of you though to don't use Gene Network 1 anymore. You have to use Gemma for the mapping, Enkhe. Lulu can show you how to do that. So I noticed that you were using Haley-Knott equations. Those
really? This is,
computer mapping get better the model in the Gene Network.
Okay. So yeah, this is, data that we did last year. So we couldn't, yet update all this. So
I think
go ahead, Wenjing.
I think, aging, you can study with high-fat diet, like doing time points where you have the pups are just like 5 weeks, and as they continue to grow into adult mouse and further, you can choose like few time points and look at the, in addition to aging, like necrosis, apoptosis, and the mechanism related to muscle and heart crosstalk. So, I mean, from there, many projects can be done.
Yeah, that's the we are looking for with the high-fat diet. So for our genotypes, like for our genetically modified animals, we have standard, let's say, setting, do the echocardiography in 6 weeks, 12 weeks, 14 to 24 weeks, and 1 year and 1 year half. So this is kind of like for establishing, let's say, the time points, and use the data across all the genetically modified animals in our lab. So this is very doable, and I think we can get the very interesting data on the high-fat diet as well.
Got to sign off. Thanks, Enkhe.
Okay,
thank you very much.
Hang in there. If anybody else wants to talk to Enkhe, though, please stay.
Okay, any question? if no question, we can stop here. Yeah.
Okay,
Thank you very much.
so much.
It's great
Thank you.
talk. Yeah. Thank
Thank
you.
you everybody for
Okay. Bye.
coming. Thank you. Bye-bye.